Hello and welcome back to the Biochemistry for Health Sciences channel. In the previous videos, we talked about Main Street, perhaps the most important metabolic highway in our body. And this is the highway that converts glucose all the way down, eventually providing ATP or energy to the cell. We discussed in the previous videos the glycolysis part of Main Street, the pyruvate dehydrogenase or the PDH part of Main Street, the Krebs cycle or the citric acid cycle or the tricarboxylic acid cycle part of Main Street, and we also discussed the electron transport chain part of Main Street. Today we shall focus on oxidative phosphorylation. So we will look at the OP part of Main Street. The key question we will try to answer today is this: how is ATP, which is our major source of energy, this is how we use energy in the form of this chemical ATP and also in other nucleotide forms like GTP, but ATP is the most predominant form. So how do we make this ATP as electrons flow from NADH and FADH2 to oxygen along the electron transport chain? So NADH and FADH2 are the two carriers of electrons. They will transport these electrons to oxygen, and as these electrons flow, the question is, how does this electron flow couple to this formation of ATP? So the answer to this question is the chemiosmotic theory. So basically the chemiosmotic theory tells us how electron flow to the electron transport chain causes ATP synthesis by this enzyme here that is called ATP synthase. So let's look at the key features of the chemiosmotic theory that will help us answer this question. So just to remind you, as we discussed in the previous video, electrons from NADH as well as FADH2 are eventually accepted by oxygen, and these electrons travel within the complexes as well as between the complexes based on the redox potentials. So let's see how, as these electrons travel to oxygen from NADH or from FADH2, how this enzyme couples the electron flow to the synthesis of ATP. NADH will send its electrons to coenzyme Q. So you can see two electrons will be transferred from NADH to coenzyme Q. So we see that in this equation here, NADH is giving electrons to coenzyme Q. The NADH becomes NAD, and the coenzyme Q takes the electrons and becomes reduced to COQH2, reduced coenzyme Q. So this transfer of electrons from NADH to coenzyme Q is catalyzed by complex 1. At the same time as these electrons are being transferred from NADH to coenzyme Q through complex 1, the complex 1 also takes 4 protons from the matrix side and sends them out into the intermembrane space. How does this happen? So as NADH gives its electrons to complex 1, these electrons will travel through the various redox center of complex 1. So there is electron transfer within redox centers of complex 1. And this transfer of electrons, these redox reactions, will cause the redox state of complex 1 to change. And this change in the redox state of complex 1 will cause a conformational change in complex 1. So basically the shape of complex 1 will change. So when the shape changes, the pK values of certain ionizable amino acids of complex 1, their environment changes, therefore these pK values are going to change. So this change in pK values of the groups that are facing the matrix side as well as the groups of complex 1 that are facing the space, these pK values change, and that causes the protons to basically hop along hydrogen bonded groups, what we call the proton wire within complex 1. So the protons basically are transferred along the proton wire and enter the space from the matrix. So from the matrix, these protons enter the space. So basically, four protons are pumped for every 2 electrons that are transferred from NADH to coenzyme Q. So complex 1 not only catalyzes transfer of electrons from NADH to coenzyme Q, but is also working as a proton pump, and it pumps protons, 4 protons, from the matrix side into the space for every 2 electrons that are transferred from NADH to coenzyme Q. Recall from the Krebs cycle that when succinate is converted to fumarate, FAD is reduced to FADH2. Now complex 2 catalyzes the transfer of electrons from FADH2 to coenzyme Q. So basically the electrons are going to go from FADH2 to coenzyme Q. In the process, FADH2 becomes oxidized to FAD. Coenzyme Q with its electrons becomes reduced to coenzyme QH2. Note that unlike complex 1, complex 2 is not a proton pump. Okay, so complex 2 does not pump protons. It simply transfers electrons from FADH2 that is formed in the Krebs cycle to coenzyme Q. We shall see in future videos that FADH2 is also formed in other metabolic reactions. For example, in beta oxidation or fatty acid catabolism, FADH2 is formed by the enzyme acyl-CoA dehydrogenase. In the GPS shuttle, FADH2 is formed by the enzyme glycerol 3-phosphate dehydrogenase. So in all these cases, coenzyme Q is the acceptor of these electrons that come from FADH2. Okay, so FADH2 will send its electrons to coenzyme Q. It will bypass complex 1. So now the electrons are with coenzyme Q. So the electrons now are with coenzyme Q. Now coenzyme Q will send its electrons to cytochrome C. So coenzyme Q is going to transfer its electrons to cytochrome C. In the process, coenzyme Q becomes oxidized, and cytochrome C, you can see the ferric form, which is the oxidized form, is going to become reduced, the ferrous form, Fe2+. So cytochrome C is going to be reduced, coenzyme Q is going to be oxidized. So this transfer of electrons from coenzyme Q to cytochrome C, this is catalyzed by complex number 3. So this transfer of electrons from coenzyme Q to cytochrome C occurs in 2 steps because cytochrome C can only accept 1 electron at a time, while coenzyme Q can donate 2 electrons at a time. Now complex 3, just like complex 1, not only catalyzes transfer of electrons, in this case coenzyme Q to cytochrome C, but complex 3 also pumps protons. It pumps 4 protons for a pair of electrons. So complex 3 is also a proton pump. This proton transfer from the matrix to the space is carried out by the help of coenzyme Q. Okay, what we call the Q cycle. So the coenzyme Q also helps complex number 3 to pump 4 protons into the space for every pair of electrons that is transferred from coenzyme Q to cytochrome C. So now the electrons are with cytochrome C. In the next step, these electrons will travel from cytochrome C to oxygen, which is the final acceptor of electrons. So basically the electrons in cytochrome C will be transferred to oxygen. In the process, oxygen becomes water and the cytochrome C gets oxidized into the ferric form. This redox reaction requires protons, and these protons come from the matrix. So basically 4 protons from the matrix are used up to help form water. Now complex 4, just like complex 3 and complex 1, not only helps catalyze the transfer of electrons, in this case from cytochrome C to oxygen, but it's also a proton pump. It is also a proton pump. Just like complex 1, protons are transported from the matrix side into the space by proton wires that are present in complex 4. Now looking at this equation, we can see that for every 4 electrons that are transferred from cytochrome C to oxygen, a total of 8 protons in the matrix are used up. 4 of these protons are used to make water, and the other 4 protons are transported into the intermembrane space. Okay, so for 4 electron transfer, we have 4 protons that are being transferred from the matrix into the space. So for 4 electron transfer, we have 4 protons pumped across the membrane. Therefore, for a pair of electrons, for 2 electrons, we will have 2 protons pumped across the membrane. So that's where this number 2 comes from. So unlike complex 3, which pumps 4 protons per pair of electrons, and complex 1 that pumps 4 protons from a transfer of pair of electrons, complex 4 only pumps 2 protons from a pair of electron transfer. Okay, so to summarize, complex 1, complex 3, and complex 4 pump protons from the matrix into the space, intermembrane space. Okay, so we have protons that are coming from the matrix into the space. So this means that the proton concentration in the space, so the proton concentration in the space is going to be much more than the matrix, much higher than the proton concentration in the matrix. Now a higher proton concentration in the space, means a lower pH, because remember pH is equal to minus log of proton concentration. So the higher the proton concentration, the lower the pH. So the intermembrane space actually has a pH that is lower, 7, pH of 7, while the matrix has a higher pH of 8. In addition, protons are positively charged. That means the space where there are a lot of protons will have a net positive charge compared to the matrix, which will have a negative charge compared to the space. So basically this means that the electron transport from NADH as well as FADH2 to oxygen has caused the pumping of protons and across the inner membrane, and that pumping of protons leads to the formation of a proton gradient where the concentration of protons is very high in the space and much lower in the matrix. Now this proton gradient is associated with energy. This proton gradient energy can be split into two parts. There is energy from the difference in pH, and then there is the energy from difference in the charge. So there is a pH energy, and there is an energy from the difference in the charge between the matrix and the space. So the energy that is required to transport a proton from the matrix into the space can be calculated by using this formula here. So we can calculate delta G, the energy that is associated with pumping a proton. 2.3, R is 8.32, temperature is 273 plus 37, body temperature is 37. The difference in pH, 8 and 7, so 8 minus 7, plus this part here that's due to the membrane potential. The charge, Z is the charge. The charge for proton is 1. A proton has a charge of 1. The Faraday's constant F has this value here, and the difference, delta psi, the difference in voltage between the matrix and the space is 0.168 volts. So we can plug in all these numbers and we get about 22 kilojoules per mole. Okay, so pumping of a proton out of the matrix requires about 22 kilojoules of energy. And this energy that is required for pumping protons out came from the redox energy. So as electrons were transferred from NADH to FADH2 to oxygen, that gives off a lot of redox energy. So basically the redox energy has been converted into this proton gradient energy. Okay, great. So as electrons were transported from NADH to, and as well as FADH2 to oxygen, there was a lot of redox energy generated. That redox energy has been converted into proton gradient energy. That's wonderful, but now it still doesn't answer the question, how is all this connected to the synthesis of ATP? How is this energy that is available from these protons, how is this energy used to make ATP? And the answer to the question is that these protons that are in the space, this large amount of protons, high concentration of protons in the space compared to the matrix, they want to go back, right? They want to go back to the matrix, but they cannot because the inner mitochondrial membrane is impermeable to these protons, doesn't allow the protons to go back. However, there is one way by which the protons can go back into this matrix, and that is through this channel here called F0. So it is a F0 channel of the enzyme ATP synthase, and these protons can now enter. They can enter this channel here. So as these protons rush into this F0 channel, what happens is this enzyme starts moving like a rotational motor, and that causes the F1 part to change shape, and the F1 part, by changing shapes, is going to catalyze the synthesis of ATP from ADP and PI. So let's look at this part here in a little bit more details. So to make ATP, this enzyme ATP synthase requires the two substrates ADP and PI. Now these two substrates, ADP as well as PI, are brought into the mitochondrial matrix. So here's your ATP synthase. So ADP enters the mitochondrial matrix by a protein called ADP-ATP carrier. PI, inorganic phosphate, enters the matrix by a mitochondrial phosphate carrier. Once ATP is formed, ATP will be sent out. Some ATP, of course, will be used by the mitochondria, and the rest of the ATP will be sent out by the same ADP-ATP carrier. Okay, so now this ATP synthase has everything it needs to make ATP. It has its ADP, it has its inorganic phosphate. So let's look at how it goes about making ATP. So let's see how ATP synthase uses the proton gradient to drive ATP synthesis. Okay, we shall focus on the concepts here rather than the details. First, a little bit about the structure of ATP synthase. This enzyme has four major parts. The first part is the catalytic headpiece. So this is the catalytic headpiece. So there are three sites here that can catalyze this reaction, and the reaction is ADP plus PI gives you ATP. Okay, so three sites here that can make ATP. That's called the catalytic headpiece. The second part is the central rotor. This is our central rotor, and it consists of these subunits. And as we'll see in a few minutes, the central rotor can rotate. Okay, so you can see this rotor. It can actually rotate. The third part is the proton channel. And this is composed of the A subunit as well as the C ring. So the A subunit and the C ring. Note that the proton channel has a proton entry channel. So here is the proton entry channel, and through this channel protons from the space can enter the enzyme through the proton entry channel. And then there is a proton exit channel nearby. There is a proton exit channel, and protons from the enzyme now can leave the enzyme and enter the matrix via the proton exit channel. Also note that the C ring has multiple helices. So this is your C ring here. It has multiple helices, several helices, and we shall see in a few seconds. These helices can exist in the protonated form, where protons combine to them, or the helix can exist in the unprotonated form. So these helices play a very important role in the mechanism of this enzyme, and we shall see that in a few seconds. And the fourth part of this enzyme is the peripheral stator. This consists of these subunits, OSCP, D, F6, and the B. And basically, this peripheral stator links the headpiece with the proton channel. So these are the four parts of the ATP synthase. So now that we have an idea of a little bit about the structure of ATP synthase, let's now see how this enzyme works. Let's first focus around this area between the exit proton channel and the entry proton channel. So the story begins right here, in this region here, between the entry and the exit proton channels of this AC subunits. So here we have a helix, and this helix is next to the exit channel, and the exit channel is the channel that is facing the matrix. And remember, the matrix, the proton concentration is very low. So this helix is going to be unprotonated. Whatever protons it had before, when it reaches here around this exit channel, it's going to lose its protons and become unprotonated. So this is going to be unprotonated. So as the C ring rotates, this unprotonated helix now faces the entry channel. Now the entry channel has a lot of protons coming in from the space. These protons are going to combine with this helix and make the helix into protonated helix. Okay, so the helix goes from unprotonated to protonated. Now remember from the videos on proteins, when there are a lot of amino acid R groups, they can have CO minus. So when a carboxyl is in a CO minus form and it becomes protonated, then you get COOH. So it's a big difference between COO minus and COOH. You can see CO minus has a negative charge, COOH has a zero charge. CO minus is more polar, COOH is more non-polar because it doesn't have a charge. Okay, so there are big differences that is caused by protonation of these groups. In a similar fashion, an amine group which is unprotonated, when it becomes protonated, it becomes NH3 plus, and you can see a big difference once again, neutral here, positively charged here. So basically the concept is the protonation of the helix causes a lot of groups to change. So this protonation will affect the weak forces that is holding the helix together. And this is going to cause a shape change of the helix. So you can see the helix is shown here vertical. Here it is shown at an angle. So because of this shape change, the helix is now pushing against the A subunit. You can see it is pushing against the A subunit. And when it pushes against the A subunit, this is going to cause the C ring to move a little bit. Okay, so there's a slight rotation of the C ring as the helix changes from this shape to this shape and causes pushing against the A subunit. So this process is repeated as more helices change from the unprotonated to the protonated form and cause pushing against the A subunit, and that leads to a continuous rotation of the C ring as protons enter the enzyme. Now the rotation of the C ring is going to rotate the central rotor. So now this central rotor is going to also start rotating. The central rotor is closely associated with the catalytic headpiece. The catalytic headpiece has 3 catalytic sites. So there are 3 catalytic sites. Let us just look at one of these sites. So you can see this site here. We'll just pick one site. This site is shown in its loose conformation or L conformation. So this is the loose conformation. And in this conformation, this site binds ADP and PI very well. So we have ADP and PI, inorganic phosphate and ADP, come and bind to the loose conformation site of this enzyme. Now the rotation of the central rotor, this rotation of the central rotor here that is rotating, forces or causes this L conformation to change into a T conformation or tight conformation. And in this T conformation, the ADP and PI are forced to come very close to one another to form ATP. So remember, ADP is negatively charged and so is PI. PI is also negatively charged. So to try to bring this ADP and PI close to one another, it only happens in this T conformation, tight conformation, where these two are brought very close to one another and then they are forced to react to form ATP. So ATP is formed in the T conformation. Further rotation of the central rotor now changes the conformation from the T side to the open or O side. And this side now allows the ATP that is formed to escape the enzyme as free ATP. So to summarize, the proton flow, the free energy from proton flow, is basically changing the conformation of the catalytic site from L to T to O in order to catalyze ATP synthesis from ADP and PI. This is called the binding chains mechanism. Okay, so here we have the most important highway explained. So this is how we generate, or how your cells generate ATP from glucose. if the cell has mitochondria and if the cell has oxygen. So this oxidative phosphorylation is the most important source of ATP for cells that have mitochondria and oxygen. Of course, if you remember, that excludes red blood cells because red blood cells do not have mitochondria. Okay, so please keep this pathway, this main street, very important pathway at the back of your mind as we go through more videos on metabolism. So until the next video, goodbye and have a wonderful day. Hopefully we'll see you soon in the next video.